as a side project. I wasn't even funded to do it, to be honest. It was like a little side project I was doing with some startup funds, but everything was working. Like, I was using different drugs, which had variability in responses, but the ketones seemed to be working on all the different cell model systems. So that steered me into a whole new direction.
Welcome back to another episode of the Smarter Not Harder podcast, your home to life's 1 cent solutions to $64,000 questions. My name is Dr. Scott Sherr, and I'm your host again today. Every week, with a rotating group of a high council of hosts, including yours truly, we interview top experts in health, movement, psychedelics, fun pharma, enlightenment, performance, and much more. Before we get started, don't forget to hit the like and subscribe button below so you don't miss an episode. This episode is brought to you by Transcriptions, makers of precision-dosed pharmaceutical-grade and physician-formulated buccal troches, as well as the nonprofit organization Health Optimization Medicine and Practice, a foundational way of practicing health rather than treating disease. My guest on the podcast this week is Dr. Dominic D'Agostino. I had so much fun speaking to Dr. Dom on this podcast. I've known him for many years, and we've had a lot of intersections over that time frame, especially in the world of hyperbaric oxygen therapy, but much more than that. And we dove into a lot of different topics here today, including how Manta Rays are ketogenic, interestingly enough, Dom's early research in the field of oxidative stress, ketones in cancer, ketones and how they affect mitochondrial function both acutely and in the long term, ketones and epigenetics, and how ketones shift epigenetics in various ways, how Dom's thinking about ketones has changed over the years, counter-regulatory responses of insulin and ketones and longevity, metabolomic testing, ketones in GABA, as well as adenosine levels, and much, much more. I really hope you enjoy this podcast. We had so much fun recording it. And without further ado, here is Dr. Dominic D'Agostino. Dom, how are you?
I'm doing very well. How are you, Scott?
I'm doing great. This is really great. Thanks so much for being here and being a part of our Smarter Not Harder podcast network that we've just started. And we're so excited to have you on to chat about all the things that you're an expert in, which is many things. Of course, you're well known for the world of the ketogenic diet, but I want to start with Manta Rays because we were talking about Manta Rays just a minute ago. And I didn't know that Manta Rays actually are ketogenic. Can you tell me a little bit about your experiments with Manta Rays before we get started on humans?
Sure. Yeah, you had, I think that was, came off of, you asked me where my wife was. So she's filming a documentary in Hawaii, and I won't say any more about that because that's kind of a secret project. But yeah,
Ooh, okay.
she discovered that manta rays have the largest brains of all fish. So it became interesting and asking the question as we travel around, do research all over the place. There's some areas where they kill them illegally for Chinese medicine. So she was in an area where she was able to acquire blood and some tissue. So we looked at their ketones, of course, and their blood. They basically have a GK index of 1, glucose ketone index of 1. Their glucose runs about 50 or 60, and then the ketone levels in millimolar, that's like below 3, and their ketones are about 2.5 or 2.7 in the ones that we measured, and that's remarkable. We measured lactate too, and that was elevated. So they're basically hybrid fuel engines, and she discovered they have the largest brains of all fish. So it's kind of interesting that they're able to kind of use these multi-fueling systems for the brains. And they're very smart animals. So they that's the cornerstone of the research that she does in showing that they're quite a bit smarter than regular fish.
And so is it common for fish to use ketones as fuel? Do we know this or do we not know this?
Under certain contexts, they do. There was a lot more on lactate, because fish will be hypoxic and they can go down into the levels. And manta rays can dive down to like 2,000 meters, I think. I mean, like some crazy, depths. Wow. And they put sensors on them and they can go up and down, and they have these really interesting dive profiles. And their brains are astronomically large. They're almost like an alien because they just don't fit the evolutionary branch where they came from. And like a whale shark's brain is like the size of a golf ball. And a manta ray brain is like almost the size of our brains. Like we have one in our refrigerator. It's like that big, so they're huge brains. it's interesting to see how that they can maintain that brain metabolism under hypoxic conditions. And I think there's a lot
Hmm.
that we can learn. It's called biomimetics, and understanding,
Mm.
how fish move through the water and things like that, but also understanding how they extract oxygen, how their metabolism works.
Hmm.
It would be interesting to do like a high level metabolomics on the brain to really figure
Mm.
out what's going on there.
Yeah, I mean, it's interesting because I know you got a lot of your start in the world of diving
Mhm.
and the world of understanding Navy SEALs and what they were doing
Yep.
underwater. And that's how you came, if I'm not mistaken, to the ketogenic diet as a way to prevent oxidative stress. Is that right?
Yeah. Well, my postdoc started in 2004, and it was basically measuring reactive oxygen species in high
Mm.
in these pressure chambers that were developed in Ohio, where we could do patch clamp electrophysiology and fluorescence imaging,
Hmm.
and ultimately atomic force microscopy. And we
Hmm.
observed like big increases in reactive oxygen species. So I was using SOD and catalase mimetics to prevent that. And the current theory of oxygen toxicity posits that it's largely response. It's a response to reactive oxygen species overproduction from an increase in brain PO2 levels that get really, really high.
Hmm.
The first strategy was to sort of attenuate that ROS increase. But the more I looked into and I looked into lactate and other things, it became more interesting to me to target energy metabolism, because the
Hmm.
energy through the mitochondria is what was generating the reactive oxygen species. Based on, I looked at NADPH oxidase and xanthine oxidase and all these different things, but our probes that we were using, fluorescent probes, determined that you could see the mitochondria were basically pumping out superoxide anion. So I used different energy systems like glucose and lactate and different, 2-deoxyglucose to block glycolytic activity.
Hmm.
And the work from Zhong Ro, who was at Barrow Neurological Institute at the time, showed a pretty sharp reduction in reactive oxygen species in model systems. So I started tinkering with these things, but only in cell culture, and saw
Hmm.
in the presence of 2 millimolar beta hydroxybutyrate, we could pressurize the chamber to 3, 4, 5 atmospheres of oxygen,
Mm.
and it was really knocking down the reactive oxygen species. And in further studies, like we have a little pressure chamber that we can go real high. The oxygen would kill the cells, but in high ketones, the cells would live. So it became even more
Mm.
interesting. It's like, well, this is something that I not have to study. Also, because the ketogenic diet is used for drug-resistant seizures, and oxygen
Right.
toxicity is seizures by definition. And
Right.
many of the drugs, the anticonvulsant drugs, are largely not very effective. Yeah, they are, but in really high concentrations where you would sedate the
Right.
subject and things like that. So, yeah, and
Sure.
that's
Especially the older ones.
yeah. So it
Yeah.
took about, the study started in like 2004, but it wasn't until about 2008 that I really started doing what I would call ketogenic studies, where we were like starting
Got
to
it.
formulate them for rodent model studies. I was tinkering around as a side project. I wasn't even funded to do it, to be honest. It was like a little side project I was doing with some startup funds. Um, but everything was working. Like, I was using different drugs, which had variability in responses, but the ketones seemed to be working on all the different cell model systems. So that steered me into a whole new direction.
Mm. And then was it through that work with the cell cultures and understanding that the ketogenic, these molecules, the beta hydroxybutyrate, were potentially effective at mitigating oxidative stress, that you gravitated towards looking at other ways you could use these molecules to prevent oxidative stress in other realms, other types of areas? I know cancer
Mm-hmm.
probably came later,
Yeah.
but I think before that was some other steps along the road. Am I not? I think
Yeah,
correct,
yeah.
right?
So I was looking at beta hydroxybutyrate, and then um, basically I just wanted to get a methods paper out to demonstrate hyperbaric atomic force microscopy in different cell
Mm.
cultures. Like I had smooth muscle cells, human fibroblasts, cancer cells, neurons at the hippocampus and cortex. And it was like making all these measurements, and it was like, yeah, let's put a fancy paper together on hyperbaric atomic force microscopy, right? And in the process of
Sounds cool.
yeah, in the process of doing that, we had a cancer cell line at U87 glioblastoma cell line and a C6, I
Yes.
think. And when we hit them with oxygen, we noticed that they died faster. So, which is very interesting. They died like much quicker than the normal healthy cells, the cancer cells did.
Mm-hmm.
So it gave us the idea. And then I started looking at, chemotherapy drugs. Many of them kill cancer through a redox mechanism.
Yeah.
And then I connected with people at Moffitt Cancer Center who had a background in hyperbaric oxygen. Actually, Dr. Robert Gatenby. He's like a world-renowned cancer biologist, mathematician, and a biophysicist, and did some work with hyperbarics. And he told me about the efficacy of radiation is proportional to the PO2 of the tumor. And when you reverse tumor hypoxia and hyperoxygenate the tumor, you can even remove the person from hyperbaric oxygen. The reactive oxygen species will stay elevated for like 45 minutes to up to 2 hours later,
Mm-hmm.
and the hyperbaric oxygen would be more effective. So we started developing protocols. One was to use a chlorotoxin conjugated radioactive nanoparticle that you would administer, and then the chlorotoxin would find the GBM and carry the radioactive nanoparticle. The radiation would decay in like a couple hours, but then you would use hyperbaric oxygen during that time. So we started developing these proposals. Nothing got funded, but I kept doing the work in the lab, and it was very consistently I would see more cancer death. And then I started treating the cancer cells with ketones, and it would decrease their proliferation. It would decrease their proliferation, but also we saw more dead cells. And I was like, this is interesting. So then I have med students that come into the lab, and pre-med students, and they try to get some research for their application.
Sure.
And there was like 3 students that reproduced the results. And then I had a student come along, a PhD student, and she made it her PhD dissertation project. And that was Angela Poff. So now Dr. Angela Poff. And
Yes.
and she did some beautiful work in the lab with hyperbarics. Basically, the synergy between the ketogenic diet and hyperbaric oxygen in a very unique model of metastatic cancer. And she did a lot of cell culture work too. So yeah,
Right. Yeah. And that's how I got to know you. And that's a good background from, I first learned about your papers coming out with Angela and hyperbaric oxygen therapy, combining it with the ketogenic diet for the model of glioblastoma that you're describing. And this must have been back in 2013 or 2012, I think.
Yeah.
When did it come out? It's somewhere around that time,
I think,
right? Or was it earlier?
we had that. Well, we did the study. Right? I think the study was done like early 2012, but it was, the reviewers were like, "What's this?" It's kind of we were pitching it to different. I think PLOS One was the one that like picked it up, but they had trouble finding reviewers that were suitable and knowledgeable enough to there was like, this is a well written paper, but we just can't find reviewers to look. Or some of the feedback too. I think there was some feedback with the ketogenic diet, and this is a very good point. We had to go back and do more experiments because they said with the ketogenic diet, you're inadvertently calorie restricting, and we know that calorie restriction can reduce cancer growth over time. So we had to go back, and it was a good reviewer comment to go back and do another group, a control study where we took the standard diet, calorie restrict it to weight match the ketogenic diet. And then we did see an effect with calorie restriction, but it was not the same. We saw a much bigger effect with the ketogenic diet, which was not calorie restricted. It was ad libitum. And I think after we had to go back and do those control experiments, it got accepted.
Got it. Well, that's really interesting. I mean, especially because we know that, excuse me, on the ketogenic diet, people tend to eat less calories in general, right? They eat less because the food is more dense. But I know that in further studies, you were looking at the ketogenic diet sort of ad libitum versus a restricted ketogenic diet as well, where it was calorically restricted. So this is what you mean when you were comparing the two together, as opposed to having it just ad libitum, correct?
Yeah, well, my colleague, Dr. Thomas Seyfried, who really inspired this research, was very adamant that the diet should be calorie restricted. And to do calorie restriction in mice, you have to weigh out the food, then you give it to them, and then they eat it all within an hour, and then they basically fast 23 hours. Sure. And kind of, it involves more oversight. So we were I developed a I developed a diet that I called the protein sparing modified ketogenic diet. So you've heard of the protein sparing modified
Mm,
fast. So a protein
sure, of
sparing
course.
modified
Yeah.
ketogenic diet was higher in omega 3s and it was higher in protein. Instead of like 10%, I doubled it to 20%, and I put MCTs in it. So it was a ketogenic diet with modified ketogenic diet macro ratios, but a large percentage of the fat was MCTs that would further elevate the ketones,
Mm,
and it had the higher protein. And that diet was, that we got really nice results from that diet, and I
mm,
worked with
mhm.
a rodent, manufacturer, and we went back and forth on developing, the optimal diet. And, that was optimal at the time. I would change things now, but the higher protein, I think,
Of course.
was
Yeah.
beneficial for the cancer cachexia and I think the immune system and things like that. Um,
Right.
yeah.
Right. So you were involved in obviously the cancer
Mm-hmm.
world and working on some of that and starting to get some grants and writing papers. You're also involved in, I think, the Office of Navy Research and working with divers around that time. How did you get involved? It's just funny because the first time that I saw you on a podcast was with Dave Asprey, and this is like podcast number 40 or number 50 or something like that for him. It was
Yeah.
very early on, maybe 2013.
Yeah.
How did you get involved in the sort of optimal performance world, and why do you think that started becoming such a like a hot topic back in, like, that was relatively early, but it obviously has become very much more interesting and much more involved in the whole physical performance, optimal performance world then or now. But how did you start getting involved in this world? Was it some of the research that you was doing, or some of the just interest in you that you had personally, that like in the biohacking optimal performance world, that kind of got you now involved in that side of things as well?
Yeah, I had no real interest in that world or knowledge of the world of podcasts and stuff. I had done some small well, no, I guess they were bigger because it was like NPR, like, podcasts and some public radio kind of podcasts and things. Oh, and then
Sure.
probably the big one that kind of put me on the map was the Mercola podcast. So Joe Mercola. So
Sure.
he had
Yeah.
me on,
Of
and
course.
I had not really done many podcasts, and that was maybe 2011 or 12. But like
Okay.
24 hours after that podcast, it had something like almost 500,000 views. It was like crazy.
Wow.
It was or maybe a day or two
Yeah.
after, it was like a 500,000 views, which was, somehow it got shared around and kind of went viral. And then I started getting a lot of people reached out to me to discuss more about that particular study. And then they just found out about the research I was doing. But I was, your
Yeah. It was.
typical introverted scientist just buried in the lab 24/7, working every weekend, running experiments, analyzing
Yep.
data, that kind of thing. So,
Yeah, I mean, I remember you being on the podcast with Dave
yeah.
Asprey and talking about the ketogenic diet and talking about it for performance and in the ways it could be used in sort of in a very different way than you and I had initially spoken about it when we talked about it
Hmm.
with cancer and combining with hyperbaric oxygen therapy, which is my specialty, right?
Yeah.
So I was working in clinics in San Francisco using hyperbaric therapy, and people were starting to come in my door asking about, what can I do this with the ketogenic diet? And back in 2013, there was very few resources out there except for your website, which was
Yeah.
nascent
Yeah.
at the time, was
Yeah. Yeah.
ketonutrition.org, right? So, and I know
The
that's still
skeleton
around now.
website. Yeah.
And yeah, yeah. So I would send people there, but now obviously there's like a huge amount of resources
Hmm.
for people, which is just kind of a testament to the work that you've done. And part of the work that we're doing at our nonprofit, Health Optimization Medicine and Practice, is trying to educate doctors and practitioners on a new way to
Hmm.
practice health and to optimize health rather than treat
Hmm.
disease. And obviously we know that the ketogenic diet seems to have some significant potential from a longevity perspective, potentially also from just a quality of life perspective. And one of the big things that we talk about a lot is
Hmm.
metabolomics, which is the study of metabolites, the study of what I like to call like the real-time cellular mechanisms that are ongoing right now. And I know we talked about that earlier with the Manta Rays,
Hmm.
and I know that's a lot of the early work that you did in the lab as well, to kind of understand what was happening. And beta hydroxybutyrate obviously being a
Yeah.
metabolite, and then other ketogenic acetoacetate, others of these are metabolites that you can measure. One of the things that I find interesting is how the ketogenic diet or being in ketosis in general affects your mitochondria specifically. Now, we know there's various ways of producing energy. Is there a sense that you have from the work that you've done? I mean, I guess the first question is how does it initially impact mitochondrial function once you switch over to the ketogenic diet or being in ketosis? And is there a long-term benefit from being in ketosis over the long term for mitochondrial function that we know of?
Yeah, good questions. And I think it's an area that's still a nascent area of research. But what I'm confident about, commenting on this is that when you initiate a ketogenic diet that's carbohydrate restricted, suppresses the hormone insulin, which further decreases glycolytic activity. The decrease in ATP production, oxidative phosphorylation contributes to about 80 to maybe 85% of the cell's energy, mitochondria. But in the context of carbohydrate restriction, glycolytic suppression through insulin suppression, it actually increases the demands on the mitochondria become higher. And that's hormetic stress, if you want to use that term. The mitochondria have to work harder, and in the process of doing that, you might get a burst in reactive oxygen species or an elevation, which is part of the sort of stress response. And then you activate a number of pathways that are associated with mitochondrial biogenesis. And much of this has been written about and talked about in discussions or papers by Mark Madsen. His work on caloric restriction and fasting got me interested in mitochondrial biogenesis and things like that. So the mitochondria then adapt over time and upregulate fatty acid oxidation. So beta oxidation of fatty acids become elevated through the mitochondria, then generates more ATP through fats. Because if you're fasting, fats, or the ketogenic diet, which is basically switching your body to more of a fat fuel source. And then accelerated beta oxidation of fatty acids in the liver will contribute to ketone production that will then not be used as an energy source in the liver, actually, because the liver lacks succinyl-CoA transferase, which is required for ketone oxidation for energy, but it spills into circulation. So peripheral tissues, namely the heart and the brain, especially the brain, central nervous system, relies quite heavily on ketone metabolism in the face of starvation, in the face of fasting, and in the context of the ketogenic diet too. So, and this is all really mitochondrial driven process. So the mitochondria then need to work quite a bit harder. There's, mitochondrial biogenesis that occurs. I don't know if we know the brain, but it definitely occurs. I think some studies, rodent studies in the brain, but in the muscle, in the heart and in different organs, there's an increase in the density of the mitochondria and their function in general. So in the beginning, some reports are like the ketogenic diet increases free radical production. But if you kind of look at all the studies as a whole, what happens is the adaptive response to fasting or the adaptive response to the ketogenic diet, and it's that adaptive response, I think, that's important, right? Ultimately results in an improvement and an increase in mitochondrial efficiency and an increase in mitochondrial energy production by increasing total mitochondria and by increasing the efficiency of the mitochondria. So by efficiency, we're talking about like the delta G of ATP production. It's like, so you're generating more ATP, per oxygen through the electron transport chain and producing less superoxide anion, which was one of the original observations that I made when I was adding ketones to cell culture, is that I could measure in real time superoxide production, and then I would see the slope of that production, and I would use something called dihydroethidium. And then I was using mitochondrial specific redox regulators, and I would see like a leveling off of that superoxide production. and while you're giving the cells more energy. So I would keep glucose basically stable, add ketones, and then it would
Hmm.
decrease reactive oxygen species. So this was very intriguing to me and led to me following up, actually contacting other investigators like Dr. John Rowe, who had published on this, Mark Madsen, Dr. Richard Veach, the late Dr. Richard Veach, Dr. George Cahill when he was alive.
Sure.
And, they were
Sure.
basically gave me very compelling statements that ketone metabolism is enhancing mitochondrial function, could improve, neurological. I think at that time, Dr. Madsen was, identified fasting and BDNF, and then beta hydroxybutyrate he later published was the increase in BDNF was associated with the fasting induced increase in beta hydroxybutyrate. So that link there, that it was like a
Mm-hmm.
metabolite
Hmm.
associated thing. So,
Hmm.
and then
Hmm.
I started doing studies in Alzheimer's disease models and ALS. And now we do rare metabolic diseases like glucose transporter type 1 deficiency syndrome, Angelman syndrome, Kabuki
Sure.
syndrome, pump syndrome, all these different projects we have going on now. And really looking, and basically the
Wow.
whole thrust of this is enhancing energy production. Well, in the context of Kabuki syndrome, it's ketone, um, I guess you would describe it as metabolism associated epigenetic regulation. So, and then ketones,
Hmm.
the ketone metabolite beta hydroxybutyrate, can function as an epigenetic regulator through altering different
Right.
enzymes called sirtuins and NAD. And also, the PhD project that my student's working on now is actually looking at something called beta hydroxybutyrylation, where beta hydroxybutyrate can directly interact with the histone and cause histone modifications that can induce the expression of genes. In the context of Kabuki syndrome, it's been very therapeutic. So, and
Hmm.
this was work done at Johns
Wow.
Hopkins, and then they did the ketogenic diet. So we're following up with the same animal model and using exogenous ketones.
Wow. That's amazing. A couple questions there. Just to kind of double click on the epigenetic aspect of the ketogenic diet. So you mentioned that beta hydroxybutyrate is an epigenetic modifier. And so you mentioned it works on the histones and changing DNA expression through that mechanism. Are there any other mechanisms that we know that ketones work? You said sirtuins as well. Is that
Yeah,
right?
well, and I, the, this is a very nascent field, and actually, the whole
Yeah.
field
Yeah.
of beta hydroxybutyrate functioning directly on histones, it's very nascent. And I was a little hesitant for my student to do it, but it
Yeah.
looks like it's a real thing. So, yeah, going back to,
Cool.
there's different sirtuins, and I think of
Right.
sirtuins as sort of regulators of homeostasis in the cell. And I think there's seven of
Hmm.
them, and then there's like SIRT1, resveratrol. There was a big big discussion about that, whether you believe it or
Right.
not.
Right.
And actually, I think
Sure.
it does have some effects depending on the form. And SIRT3 and SIRT4 are linked to metabolism, the TCA cycle, and metabolic control and metabolic homeostasis. And then the other sirtuins play a big role in DNA repair, especially double-stranded breaks and things like
Right.
that. So,
Hmm.
and now sirtuins actually use NAD as almost like a fuel. So it's very interesting. So, and as we age, NAD levels will precipitously drop over time. Like a 40, 50-year-old has
Hmm.
like
Hmm.
40% of the NAD they had, when younger. So the ketogenic diet, and a lot of people have been trying to get, with this like overarching theory about how the ketogenic diet works. And one of them is, and this was presented at American Epilepsy Society meeting, and the data's there, is that NAD and the ketogenic diet increases NAD levels, and NAD can impact various epigenetic regulators like sirtuins,
I see.
and by virtue of elevating and sustaining higher NAD levels through ketone metabolism, um, that's affecting epigenetic regulation, and then an upregulation of superoxide dismutase and catalase, and an increase in endogenous antioxidant
Some of the antioxidants.
mechanisms. But
Right.
I don't like to point to one mechanism and say that's how it's working, because we've done enough experiments in our lab to show, and it becomes messy when you have a diet that changes so many things. So we actually do experiments where we take a standard diet, and then we formulate the standard diet by integrating the ketones into the standard diet. So it gives more confidence that what the beneficial or negative effects, and we've seen negative effects, and we could talk about that. The beneficial effects that we're seeing are ketone dependent, right? And we have seen it's just you can study drugs and the results are all over the place, but with ketones, we just see beneficial effects across the board. It's very dose dependent though. And if we elevate ketones too high, then we start to see negative effects. We start to see ketoacidosis. We start to see, negative effects. So I think, and this is, if you ask me, how has your thinking about this changed over the years? The one thing that
Sure. Please.
when we first started synthesizing ketones, I was chugging the stuff to the point where I was getting sick. And then I found out through trial, and this is way before they were available commercially, but I found out through trial and error that really just elevating your baseline level 1 to 2 millimolar is, if I was going to take a subjective questionnaire, I would reliably, point to that. If I was, determining how I feel and it would take a measurement at that point in time, it would always be between 1 and 2. And then come to find out, actually, we presented a poster basically showing that if you get ketones too high, it can make cancer cells grow. And then it can alter, and then because you're adding energy to the system and you're shifting redox control.
Okay.
So, and also if you take a large dose of a ketone ester, and you can't really achieve this with salts because it's a little bit dose limited, but a large dose of a ketone ester, if you go above 2 millimolar, then it causes a release of insulin. And that insulin can shut down your own ketone production. And what happens is, so if you're on a diet and you chug a ketone ester, which I've done many times, you feel great for about an hour or two. And then about 2 or 3 hours later, it clears out of your system and your ketones go down, but your own ketone production also goes down. And because you released insulin, that facilitates peripheral glucose disposal. So you're hypoglycemic and hypoketotic, and then the brain goes into an energy crisis. Then you get a headache and you just don't feel right. So I did this many times, but I was of the opinion that, I don't know, as it's not a negative thing. But then I started doing blood work and I realized, whoa, my ketones are out, and then my glucose gets low too, which is a very interesting phenomenon. And there's exogenous ketones lower glucose by potentially insulin, but it also enhances insulin sensitivity. And we've shown through a lot of elaborate blood work measuring insulin, and it's not real cheap to measure insulin, and I spent quite a bit of money measuring insulin, that the threshold is about 2 millimolar. So you'll never get above that with MCT, and you'll never really get above that with a ketone salt. Some products can get you right at 2 millimolar, but a ketone ester can push you over that. And we've also demonstrated if we gavage the animals with ketone ester, we've, sacrificed a number of them by accident initially, because it was putting them into ketoacidosis. So that's why I think something like a ketone ester needs to be, for me, someone my size, I'd have to consume a half of a liter to get that. But there's there's real dangers to these things that people need to consider.
Yeah. So you bring up a good question. Do we know the mechanism as to why, if you shoot ketones up high, that insulin rises? Is it a stress response? Is it a cortisol response? Or what's causing your insulin to drive up? Because as I've always thought about this as you've described it before, which is you're increasing ketones so that you're going to increase insulin sensitivity. and so that your glucose is going to drop, but your ketones are going to rise,
Yeah.
kind of thing. But what you're saying now is that if you're going too high on your ketone ester dose, which, as you said, salts are very difficult to get you up that way, that you can actually cause a spike in insulin. And do we know why that's the case?
It's very well characterized in the literature that you can get a ketone-induced increase in insulin, which is a counter-regulatory mechanism. For example, like if you fast or if you go on a ketogenic diet, beta oxidation of fatty acids contributes to ketone production. Your ketones get higher and higher and higher.
Sure.
And then you have a couple ways to eliminate them. One is ketonuria. So you'll find ketones in the urine. And in the course of a day, you could eliminate about 100 or 150 calories of ketones. So in some ways, Atkins was right. You are peeing out calories, but maybe a cookie might be like a cookie's worth of calorie. And then
it's fruity. That's the
right.
fruity urine that we know from medical school, right?
That's right.
It smells fruity and tastes fruity.
I know, that's how they used to
it
test
myself,
in the old days. They would go like this and be like, yes, type 1 diabetes, diabetic. So that's one way our body's trying to bring the ketones down. And another way is that as ketones get higher and higher and higher, the energy from the ketones kind of feed back to the liver and the pancreas and actually cause the pancreas to release small amounts of insulin. It doesn't take much, small amounts of insulin that then has an anti-lipolytic effect, right? So otherwise you would have runaway ketogenesis, which actually happens in type 1 diabetics who do not have the capacity to then have a ketone-induced release in insulin. So they just have runaway ketogenesis in the context of hyperglycemia, which exacerbates the acidosis. And then you have electrolyte disturbance and a collapse of plasma volume and a shock, right?
Yeah.
So, so that's, to answer your point, is that the body has evolved a very delicate mechanism to control ketosis, and a ketone-induced release of insulin is one of the counter-regulatory mechanisms. Yep.
I see. So to prevent somebody that has insulin capability from going into DKA, what they'll do is they'll cause a spike in insulin to mitigate the oxidation related to the ketones. That makes sense. Okay, gotcha. That's super interesting, and I think this is news for me. In the sense
nobody knows that, and maybe nobody. Just not to interrupt you, but I don't think most people know that, and I've never described it in that level of detail before in a podcast. I don't think so, and I think it's in the context now where exogenous ketones are available, the 1,3-butanediol, and I think it's important that people know this. Yeah.
That's what I mean. I think that's really why I wanted to kind of to focus on that for a minute, because as well as I do, that these are just all over the market now. People are chugging ketone esters for their performance boost, but it can have as a counter-regulatory issue that they didn't know about, and now they're going to have this brain fog for an hour or two afterwards because they have no glucose around and they have no ketones around because of what's going on. And it sounds like, I know there's probably some significant benefit from the esters, obviously, and I use these as well in my own clinical practice on the hyperbaric side, especially if we're worried about people that are under higher oxidative load, if they're getting chemotherapy, if they're getting hyperbaric oxygen therapy, et cetera, and we want to make sure that we're mitigating potentially oxidative stress. And I want to go back to oxidative stress for a minute too, because on the oxidative stress side, you were talking about some of these early experiments you did where you found that you was producing less oxidative reactive oxygen species, but actually making more energy. And so is it kind of a finite thing in the sense of, can you maintain in this way, or is it that over time, is there any issues by maintaining in ketosis that we know about so far? I mean, obviously we know that children on the spectrum of seizures have been maintained on ketogenic diets for long periods of time without any issues. I know there's a big push in the, let's call it the metabolic community, right? In the sense that we should be pushing ourselves out of ketosis, being more metabolically flexible. Do you have a sense of, from a longevity, from a health perspective, if that's a good way to go, or is it depend on the person? And some people that they could stay ketogenic, I know like you for the most part, Dom, for forever and be fine, but others need to kind of cycle out to feel like they're still optimized in the ways they want to feel.
just to back, like I've added like one carbohydrate meal in a week. So just like Sunday nights, yeah, I had like two
Nice.
burritos the other night, and then I'll
I know Scylla probably forced you, right? She's the carbaholic,
yeah,
right? If I remember
yeah. You know
correctly.
what? And her
So
biomarkers. So that's another thing we talk about. Like she got all her blood work done for like life insurance purposes or whatever. And it's like, we're comparing. It's like, man, we're like very similar. So, and even her hemoglobin A1C is really
yeah,
low. And I, I'm not going to go into because she gets mad when I talk about how much sugar she eats on a day, but she does very well. She's very carb tolerant. I wish I was.
right. So
Yeah.
that's the question, right? So like when you're doing this kind of thing, like I guess, and this goes towards your question earlier or your comment about, is there a bad side? Is there side effects? Is there potential people that shouldn't
Yep.
use the ketogenic diet, for example? Or is there certain people that should just stay on it for a short period of time if they have a specific reason? Like, I don't know, have we been able to tease this out a little bit now, or are we still kind of in that nascent
Yeah,
stage of
good
understanding it?
question. I think for long-term kind of adherence to the ketogenic diet and if it's optimal or not, or even healthy, or if it's going to kill you and give you a heart attack. I mean, we can actually just reliably point to the very well-controlled research that's being done in epilepsy patients. Or patients with glucose transporter
Sure.
deficiency or metabolic, and I'm in correspondence with people at Johns Hopkins that have had people on a classical and modified, just in a state of ketosis for 3 decades, and their blood work looks great. It's
Mm,
like, and they're on a far stricter ketogenic
right.
diet than me for like 3 decades for epilepsy and other disorders.
Right. Than most people.
So
Right.
the question I think you're asking is it optimal to stay in some state of ketosis all the time? And I don't think it's actually, I don't think it's natural, and I don't think it's optimal. I think it's optimal to go in and out. I do think it's optimal to be
Mm-hmm.
low carb. Low
Mm-hmm.
carb, my definition is like under 100 grams a day, which is quite a bit lower than the
Mm-hmm.
recommended allowance or recommendation. But to
Mm-hmm.
transition in and out of ketosis, not to do that would be unnatural. So I think at the very least, people should, do 1 week out of a month where they're at least in a mild state of ketosis with some form of carbohydrate restriction. And it could be not even carb restriction. It could
Mm-hmm.
be just doing an intermittent fasting, once or twice a week or something like that
Sure.
would get some real benefits. It does all come down to like calorie
Yeah.
management too. And there are people who believe calories don't
Okay.
matter, the calories matter. I'm not on one side or the other. I think calories matter, but maybe less than most people make it out to be. But, what I was saying is that the efficacy of the ketogenic diet and intermittent fasting for weight loss is largely due to the ability to create and adhere a
Mm-hmm.
calorie deficit. So, and that can be very beneficial in the context
Okay.
of longevity, especially for people that are problematic in regards to not optimizing metabolic biomarkers. Just simply getting your weight, getting to an ideal weight, can correct many of the metabolic uh,
Sure.
issues. I'm also very low tech when it comes. I mean, we do metabolomics, we do cytokines, chemokines, all these things in the lab. But at the same time, I like to take a step back, and I think there's like what I call functional biomarkers, right? So being able to do a deadlift with like twice your body weight for a couple reps, some push-ups, some chin-ups, and there's goals that people can set where if they just focus on function and increasing their functional output and their strength, that's probably super
Right.
reliable in regards to longevity. Like if you're 80 years old and you can do like 20 push-ups, that's like you can measure all the things in your blood
Totally.
and as biomarkers, but that's going to be crushed by the functional biomarker of being able to do 20 push-ups when you're
That's
80,
always important.
right?
Yeah.
So I think
Yeah.
these are things that people
Yeah.
need to
I think people
consider
forget those functional markers.
because
I think data is really
yeah,
important because it really does help drive
yeah,
individual decisions about what to do, in the sense that somebody on the ketogenic diet
yep,
that's micronutrient deficient and they just can't maintain, then it might
yeah,
be something that they want to transition or to change from, for example, as opposed to somebody else who looks
yep,
great on the ketogenic diet or a modified kind of ketone diet or some sort of cyclic aspect of all this, then they're doing great. Then that's great too. I think one of the things that I think we try to do with our organization is try to look at the data, right, in the sense, but not only look at the data, but look at the functional aspects, like you said, of how people are actually living and surviving and working, and their relationships, et cetera, with their environment, with people around them and things. And like you said, like grip strength is a huge one, right? Compared to if I'm looking at somebody's micronutrient levels, do I care more about their grip strength? Do I care about their micronutrient levels? It's, it's an interesting, we know that obviously that there's a lot of longevity data related to things like grip strength, and we don't have as much on the micronutrients, although we obviously think that if your micronutrient sufficient, that's going to be optimal for energy metabolism and oxidative stress and resilience, et cetera, and things like that. One more thing I wanted to ask you about on the metabolomic side was about neurotransmitters and the ketogenic diet. So do we have a sense of how neurotransmitter balance changes once somebody decides to be more ketogenic or cycle through ketogenic diet?
Yeah, so this is an area we're very interested in, and part of so we do metabolomics on serum, but we also, the advantage of doing metabolomics in rats is that you can do hippocampal brain metabolism.
You haven't done it to yourself yet? No brain surgery to yourself quite?
Not yet.
I could see you going through some like intrathecal thing at some point, I'm sure. So.
Yeah, intrathecal. I mean, that's they do that with like kids in different clinics. But I mean, we literally, we rapidly dissect out the brain and rapidly micro dissect the hippocampus, and then we flash freeze it, and then we run metabolomics on it. And so some of the insights, some of the takeaways from ketogenic therapies, and there's a ton of overlap between the ketogenic diet and exogenous ketones, is adenosine goes way up. I did not know this. I mean, this is like going back 10 years ago when we were sending stuff to the company Metabolon, and they're doing it. I'm not sure if they're even, they must be in business, but they did a pretty detailed analysis of that. And so adenosine is quite a bit higher. GABA is higher. The GABA to glutamate ratio is quite a bit higher. And that's due to an increase in glutamic acid decarboxylase. So you're converting more excitatory amino acid transporter to
so interesting, because you don't feel fatigued typically if somebody goes into the ketogenic diet, if they're in like starvation ketosis or like a fasting, they typically feel more energy and more awake and have a harder time sleeping. Do we have a sense of why that would be the case? But you have more GABA floating around.
Well, GABA is kind of interesting. I mean, if I take GABA, and I've taken it in different forms, like just GABA, you can get it at the store. And also, if I took phenibut, I haven't taken it in a while, but when I take phenibut, people take it to sleep, but I feel like almost a dopamine hit. And I think it does maybe release dopamine.
It does a little.
I actually feel more relaxed, but more energized. And I think, some people, I remember giving it to my wife, and she just slept in the next day and was like, oh, it just made her dizzy and groggy. And I was like, I don't want to take that again. But for me, it, made me feel, I don't know, like a big boost in mood and awake. Yeah. And I think it's really about neurotransmitter balance, and the anti-seizure mechanism of the ketogenic diet is probably largely linked to changes in neurotransmitter systems, GABA being one of them. The field generally agrees upon that. And some of the newer data we've published, and other people were publishing it before us, but we decided to look at it with adenosine and adenosine receptor signaling. But also we found that in the blood and also in the brain, an increase in TCA cycle intermediates like alpha ketoglutarate and succinate, fumate. And so there's a general increase in some of these TCA cycle intermediates. And, the TCA cycle generates the reduced intermediates to drive the electron transport chain. So we saw this in mice and in rats. But the neurotransmitter, the big things that jumped out, there was nothing with serotonin or dopamine that I remember, but I know GABA. And then I actually did measure neurotransmitters in myself, but I did it through a urine neurotransmitter thing. Yeah. And, if you believe in that, GABA was it was like 2 or 3 times higher than what's normal. And I was not supplementing GABA or anything. So that was very interesting. So I think we can reliably say multiple lines of, independent research has shown that GABA is elevated and adenosine is elevated. So these are undisputable
Sure.
changes that are happening with the
So,
ketogenic
I mean, I
diet.
think most people have heard of GABA before. You know, gabapentin is the drug,
Mm-hmm.
and we have GABAergic drugs
Yeah.
that we use, like the
Yeah.
benzodiazepines of the world, the Ativan, the
Yeah.
phenobarbital. These all work on the GABA system. So typically, we think of GABA as
Yeah.
a neurotransmitter that relaxes the brain, that decreases firing. So it makes sense to me that
Yeah.
the ketogenic diet, if it's transitioning more from a glutamate to GABA balance, would be preventing potential seizures, because and gabapentin is also used for seizure mitigation as well. It's just,
Mm-hmm.
it's interesting,
Yep.
because at the same time as I think you sound to be maybe an outlier, but maybe I'm wrong, that most people that go on the ketogenic diet initially feel a lot of energy and they have a hard time sleeping. And because the way I've always thought about it, and the way I describe it, is that when you're on that like third day of your fast, and you're looking for the animal on the prairie so your family doesn't die, you need to have more focus, more energy. And that's why the ketones give you that bump. So it's just interesting to me that GABA is also going up at the same time. I don't know if you have a comment on that.
Well, yeah, so it's a very good point, but I think there's many things that I did not discuss. So when we do start fasting, and I think when we go on a ketogenic diet, it increases the sympathetic nervous
Sure.
system,
Yeah.
and catecholamines too will increase. Although we didn't see it with the extended
That's good.
ketogenic
That's a
diet.
good point, though. So
And I
extended
think,
wise, you don't see that
yeah,
catecholamine release. It's just in the short term.
yeah. And I think with extended fasting, it will decrease your sympathetic nervous system activity. Actually, the GPR 109A receptor, which is a receptor to the ketone, is associated with a decrease in sympathetic tone. So, and some of our new data, we have not published this yet with some of the diving studies, is when we administered a high dose of a ketogenic agent, there's a remarkable decrease in body temperature. So we're trying to figure out what's going on here. So, the neural control of autonomic regulation can be quite complex, and thermoregulatory responses in rats are a little different than humans, but it's like one of those things that you don't need stats for to see that there's a very significant effect.
Interesting.
So we're looking into that data. We're a little hesitant to actually show the ONR because, that could be a bad thing for diving, but it's obviously a very, what I think is happening personally is that the ketogenic diet is actually suppressing sympathetic nervous system, and that could lead to some of the anxiolytic effects by reducing your sympathetic drive. Like taking a beta blocker reduces that fight or flight response. Propranolol, I think, can also decrease oxygen toxicity. So that would make some thinking. So this is like another study I want to start proposing. I know when I fasted for a period of time, my body temperature went down, my metabolism starts to shut down too. And it's actually becoming more efficient. So your metabolism becomes more efficient with the food that you give it. And then my breath hold times underwater was quite a bit longer after doing prolonged fasting. So in the beginning, though, and some people have problems sleeping when they're 24 hours into a fast, that's because there's a stress response. Like cortisol gets elevated, catecholamines gets elevated. Hey, we don't have food. We got to go out and hunt. There's an arousal state that's activated. So when I do my 3-day fast, actually, that's when I pull out the phenibut and I just take like 300 to like 500 milligrams, not even 500, usually like 300 milligrams, and I sleep like a baby. If I didn't, my sleep latency, or I just would not sleep as much. I'd probably wake up early, after 5 or 6 hours. And I remember dieting, like to get ripped, to get like cut up. And I remember doing that, like once your body fat gets to a certain level, you just start waking up earlier and earlier each day. When you get to really low levels of body fat, and it's basically going to screw up your physiology saying, hey, you got to get going and start finding some food.
The caloric aspects of things. That makes sense. Yeah, I mean, that was one of the questions I wanted to ask you was, and this dovetails well into the sort of the idea of sleep and being in ketosis. It sounds like from what you're describing, the earlier when you first transition, it's more of a sympathetic response. That GABA is floating around, but it's not as significant as far as making you feel sleepy. You're gonna feel awake for those first several days, but after transitioning, most people will have a greater balance towards the parasympathetic side, it sounds like, and be more GABAergic in general, which is super cool. One of the other cycles that I know that gets talked about a lot is the menstrual cycle when it comes to the cycle of if does that seem to change when people are on long-term ketogenic diets, or is that if they're keeping a normal weight, and otherwise does it tend to be relatively stable?
Yeah. Well, again, I point to the epilepsy literature showing that it's a variable. So with some women or girls that are on a ketogenic diet, and this is legitimately, they're on a ketogenic diet because they're being
Right,
monitored
exactly.
by the
Yeah,
neurologist and the
right.
dietitian. Some of them go out of ketosis, and then some of them go into a deeper state of ketosis.
Interesting.
So I know that's kind of not the answer like most people want, but I think what happens is that during menstruation or during the premenopause period, that may be not the optimal time to be in a state of
Mm-hmm.
ketosis. So your physiology may be a little bit more reactive to low blood
Sure,
glucose.
sure. Yeah.
So in generally speaking, I think female physiology is more reactive to changes in energy
Okay,
status.
that makes sense. Yeah,
So
women are
yeah,
complicated. Yeah, that's what I tell my wife all.
yeah, women
I
are
tell
kind of
my wife all the
complicated. Yeah.
time. But it's super interesting, because I think about all these aspects of what we do in practice and sort of the effect of ketones. And one of the things that I get a lot of questions about all the time is microbiota, in the sense of how
Mm-hmm.
the gut microbiota shift on the ketogenic diet, and or any potential complications of being on the ketogenic diet for long periods of time. And one thing that comes up a lot for people that I see is that I check for digestive capabilities, in the sense how well people are digesting fat and protein. And oftentimes I find that people that are on high fat diets, on a ketogenic diet, are usually having a difficult time digesting fat. So I often have to put them on digestive enzymes. I don't know if you have any comments
Yeah.
about microbiota, digestive issues related to being on the ketogenic diet, or long term, short term, either way.
Yeah, I'd say there's some criticism about the ketogenic diet disrupting the diversity of the
Right.
gut microbiome. Some of the literature, at least from animal models, is looking at different species like
Mm-hmm.
Akkermansia, and then Firmicutes, Bacteroides ratio, and different species that may be specific to some of the anti-seizure
Mm-hmm.
effects. Um, Data is kind of all over, but it's important to look at the ketogenic diet you're actually studying.
Sure.
And I
Yeah, that's
think
important.
a ketogenic diet that does not have fiber is more likely to disrupt and decrease the diversity of the microbiome. So fiber slows down gastric
Mm-hmm.
emptying, and I think you have better digestion of protein and better digestion, assimilation, absorption of fat too, just because of the slowing. And then the fiber contributes to short chain fatty acid
Right.
production and like butyrate, and that can fuel the colonocytes. And the fiber also, there's some resistance to fiber in the carnivore crowd, but
Right.
fiber, I mean, I teach the GI section in med phys, and I comb over the data, and there's just so much good data
I know.
when it comes to fiber. I mean, too much fiber can be a
Of
problem. It
course.
doesn't take much. So, right, like 20, 30 grams is probably enough. But for the integrity of the gastrointestinal lining of the mucosa, the tight junctions, and for when it comes to barrier function and things like that, a diet that's devoid in fiber could be problematic for some people. And
Right.
for me, I just think I've eaten a meal with and without fiber, like the same thing, and adding it to it, and it reduces my glycemic response, which probably reduces my insulin
Of course. Yeah.
response, and actually my continuous glucose monitor profile just looks better when I incorporate like leafy green vegetables in my diet. And I think there are issues with oxalates and other things in plants that could be
Sure.
anti-nutrients. But I've sort of picked things like I eat some broccoli and asparagus,
Sure.
arugula, like salad greens, things like
And
that. So I just think of it as like
yeah, yeah,
fiber is also a tool for carrying fat.
that's it. Right. So you have, and it's hard just to eat
Mm-hmm.
fat by itself. Right. So,
Yeah,
but I
that's
think,
right.
you make a really good point that a lot of times what people get into trouble with is they forget about the fiber piece. And you said it doesn't have to be a
Yep.
huge amount, but you need that fuel for your microbiota to make the short chain fatty acids, to make the beta hydroxybutyrate, not the beta hydroxybutyrate, what's it called? You said it before.
Butyrate.
The butyrate, just the butyrate, right? So I know beta-hydroxybutyrate and butyrate are very similar, right? They both work as epigenetic markers,
functions, yeah.
right? So, so I think it's really important to make that clear that that it's likely related to the types of foods that you're eating when you're on the ketogenic diet. It's not necessarily the ketogenic diet itself.
Your microbiome eats what you eat. So you can rapidly shift the composition of the microbiome by shifting your diet. And I mean, we do have issues with quite a few people contact me with small intestinal bacterial overgrowth or SIBO and say that it cured their yeast infection, their SIBO. I get so many, probably like a third of my emails are with people with gut issues where the ketogenic diet helped them. And and so, or the carnivore diet, which is devoid in fiber, it has helped many of them. And that's, that's showing that fiber is not essential, but I do think it's helpful. So if you're going to eat a big steak and, you might even think of just like vegetables as more like a garnish, it doesn't have to like fill your plate full of vegetables, but having a little bit there, I think can be helpful.
I think there's a lot of good resets. That's what we talk about, right? If you're having, if you're on the standard American diet, if you go carnivore, you're going to feel great for a period of time. If you go to ketogenic, most of the time, you're going to feel great for a period of time. But if you're not looking at some of those aspects of your macronutrients, and then also over time, the micronutrients as well, looking at data markers and just making sure that you're optimizing there, you can get in trouble over time is what it comes down to. So, go ahead.
Some days I'm carnivore. Like yesterday, I just didn't eat I just had meat yesterday. And the day before that, just eggs and fish. I've been meaning to, since I got back from Metabolic Health Summit, to go shopping to get some vegetables that I just haven't gotten. So I've been like 100% carnivore the last couple of days, and I feel great. I feel great. So, but I don't go out of my way to eliminate, plants.
I got it.
and I genuinely like eating a salad. So
one
it's usually just a nighttime thing.
Oh, I'm sorry to interrupt. I was going to say one thing on the glycemic response that you mentioned, because I think I'm interested in this, and I know that you are with wearing a CGM. Is you've also noticed, I'm sure, different ways that you eat in the sense of the order that you eat certain food. It matters, right? In the sense of what has your experience been in that capacity when you're adding in fiber or protein or fat? Like what have you noticed is the best way to kind of keep your glucose from spiking, for example, even if you eat something maybe that's not ketogenic, maybe if you're eating like, a birthday cake or something, like what have you found to be the best there?
Yeah, it's good. I was going to say this only matters in the context of eating carbs, but when I eat blueberries, if I just eat wild blueberries, I was amazed to see me spike up like 50 points eating a big bowl of wild blueberries, right? But if I mix the blueberries, and this may sound gross to some people, in like a cup of sour cream, and I add some dark cocoa powder and some stevia and stir it up, then it becomes like a chocolate mousse, at least for me.
Nice.
Then I have like no CGM response.
So
but I was experimenting with some carbs a while back where I ate I was like sweet potato, steak, and salad. And I did an experiment where I ate the sweet potato first, and like huge, even with the sweet potato, it's like huge bump up. And then I ate the salad, steak, and then the sweet potato, and it looked like two different meals.
Wow.
Where I didn't have that initial spike in the beginning.
Yeah.
Another thing to add is that when you drink alcohol with a meal, that like significantly lowers blood glucose. I don't know why. I think there's something called alcoholic ketoacidosis, and that happens because you're inhibiting gluconeogenesis.
Right. Exactly.
Alternatively, and I have not done this yet, I've been meaning to do it, is measure my insulin response to eating carbohydrates with and without alcohol, because one of my colleagues sent some papers to me showing that alcohol magnifies the insulin response. Like there's like a synergy, like alcohol and carbs, you have like double the insulin response. So I have some insulin test kits here, so I'm going to do that and see what's going on. But it's especially people in the CGM world are sending me all these things like, look how low my glucose gets after, but I have to have alcohol. Like dry farm wines, which are like no sugar, is the alcohol that I'm not drinking like sugary,
of course,
mixed drinks or anything like that, or heavy beers.
I got it.
No, it's just dry farm wines. So that's my test alcohol.
It's so interesting. I guess I always thought about it as a gluconeogenesis kind of thing, as suppressing that.
Yeah.
And I still work in the hospital part-time. I see plenty of people that come in with alcohol ketoacidosis. So I always thought that was mechanism. So yeah, let me know. That sounds super interesting. I had one more topic I wanted to ask you about if you still had a couple minutes, which
Hmm.
is the endocannabinoid system. This is the
Hmm.
natural, our body's own system that makes our natural cannabinoids. Like we call it cannabinoids because they're named after the ones from the plant. But I think there's been a
Yeah.
significant interest in this system as a kind of global regulator for multiple other systems in the body. And we used to think, for example, that the runner's high that you get were like, we call them endorphins, but we actually think these are likely now endocannabinoids that are being released.
Hmm.
I don't know if there's been any research or if you have any thoughts about how the ketogenic diet works or interacts with the endocannabinoid system at this point.
Yeah, it's a good question, and I wish I've like researched that a little bit more before jumping on because I looked at your questions right before I jumped on. But
Oh,
I do
good.
have some thoughts on that, and I know that exercise and stress in general activate the endogenous cannabinoid
Right.
system. And I mean, I'm interested. I mean, I have CBD over here in the corner, and I use it
Yeah,
occasionally, a couple nights a week for sleep, like a pharmaceutical version,
sure.
and anti-seizure effects are very real. So I would suspect maybe an increase due to when you fast or when you're on a ketogenic diet. And I don't know, I haven't searched this enough to know it, but I am of the opinion that the endocannabinoid system plays a big role in the anti-inflammatory
Right, that's what I
effects
was thinking. So
and
maybe there's a
anti-seizure
combination
effects.
or there's an interaction as like as sort of
Yeah.
an anti-inflammatory cascades that occur as a result of more natural
Yep.
endogenous endocannabinoids. I think it's super interesting. I mean, it's certainly a field that I don't know a whole lot about yet either. And I think that, from our perspective, we know that there is these natural endocannabinoids in our body, one called anandamide specifically, which is like our
Yep.
bliss neurotransmitter.
Yep.
And we know when that one hangs around longer, we feel better. So we know that CBD, for example, is one of those that allows more anandamide to be around for longer, right? And
Yep.
as a result of that, you feel better, you have more neuroprotection. As a result of that, it seems to downregulate inflammatory pathways. And I'm sure that there'll be more coming out in this realm. But yeah, it's super interesting. And I guess the last thing I want to ask you is what's coming down the pike for you? I know you've mentioned a couple different research papers that are kind of being done. Do you have anything that's going to be published soon or that's kind of on the dock in the next little while for being published that you want to talk about? Or
Yeah, a lot of different things, actually. So, well, we're doing some work on autophagy right now. Some
okay,
projects are just starting up on autophagy where we'll be measuring autophagy in response to dietary interventions. So that's kind of interesting in the context of a disease process, but we'll have wild type mice. We'll be looking at that. And, we have quite a bit of data we're still sitting on from the NASA Extreme Environment Mission operations, where we're living in a
nice.
hyperbaric environment.
Yeah, you were down there for a
Uh,
while, right? Yeah,
yeah, yeah. So, we're just getting to the point where the summer is a time where we start writing papers. So
got it.
we'll be working on that. And I think the big one that I'm working on now, which is the next call I got to get into, is the clinical trial we have ongoing right now, which is the use of continuous glucose monitoring and the Levels Health app
Sure.
as a means to influence behavior to optimize metabolic health, but also mental health. So, an area that we want to expand upon more is metabolic psychiatry. So, interestingly, like if you're treating a patient with bipolar, you give them anti seizure drugs. So, that's how you manage the manic phase. And so, there's a lot of interest on that I have in actually treating psychiatric disorders that could be depression,
Mm.
anxiety, or
Hmm.
bipolar or others with dietary interventions
Mm.
as an adjunct or as a first line therapy if someone's not responding to drug therapy. So I'm kind of working in that area, writing reviews on it, and doing a human clinical trial where we do the GAD-7 test, PHQ-9. We have a sleep questionnaire, and we have different subjective tests to assess mood. And it's really remarkable the effects that you get just through a dietary intervention.
That's really exciting. I think that
Yeah.
the metabolic aspect of psychiatry is just not existent right now in the sense of conventional care. And the
Yep.
ability to bring that in, I work with a bunch of integrative psychiatrists and doing even metabolomic testing, for example, gut testing
Yeah.
can be transformative in these people. And then I think pairing that with real-time glucose monitoring and additional testing that you're talking about it sounds like a phenomenal project. Sounds like a big project. Sounds like a lot of fun.
It's very interesting. Yeah, the gut testing is going to be really important because, and I teach about this in the medical physiology, is that there's a bidirectional comorbidity that if you have irritable bowel syndrome, you are more likely to have a psychiatric disorder.
Right.
And if you have a psychiatric disorder, you're more likely to have irritable bowel syndrome if you're diagnosed. So there's this like bidirectional comorbidity that is finally acknowledged and actually making its way into the textbooks, if you believe that. So that there is this gut-brain axis that, 10 years ago, I couldn't talk about this stuff, but
that
has become...
I mean, I didn't learn about it in medical school. I mean, it wasn't anything that I knew. And it's not even bidirectional. It's probably like tridirectional in the sense of how all the aspects of the gut are interacting with the nervous system, not just the microbiota itself, but also the gut lining, the cells within the gut lining. So it's huge. And I think that, the work that you have done to bring the ketogenic diet out into the community has just been fantastic. And in so many aspects, and I'm grateful to you, Dom, for spending some time with me and also all the years chatting with me on the phone about hyperbaric oxygen therapy and the ketogenic diet and a lot of different things that we could have talked about today. But I'm glad we focused on the aspects that I think are really interesting for people that are outside of the hyperbaric chambers, because not everybody else goes into those things. This is stuff that's going to be hitting clinical practice soon, I think, in a lot of different ways. And we're going to see it, I'm going to see it in my patients. I'm going to be working with them, metabolic flexibility, cycling off the ketogenic diet. And I know you get a lot of people that are always asking you about these things. But you have great clinicians now to refer people to, which is great. Like me, of course, and others to help work on this stuff in real time. So again, Dom, thanks for coming on and chatting with me. And this has been really, really, really great. Is there anywhere you want anybody to go to, like major resources for you and for the work that you're doing?
Yeah, well, thanks for having me, and I appreciate having this platform to speak. I know it's going to have a huge reach and reach the people who are really sort of in the trenches, treating patients and stuff too. So really appreciate the educational platform and the educational outreach you're doing by doing this interview. I guess the one-stop shop for everything we do is ketonutrition.org, ketonutrition.org. We have a blog there. We have resources. Go there, check it out, sign up for the newsletter. So we send things out in the newsletter about a month before we put it on the blog. And hopefully it's informative for people who are interested in this topic.
Awesome. Thanks a lot, man. It's been a real pleasure. I hope you enjoyed this episode with Dr. Dominic D'Agostino and yours truly, Dr. Scott Sherr. If you did, please like and subscribe below so you never miss a Smarter Not Harder podcast episode. This podcast is brought to you by Transcriptions, the makers of buccal troches that are precision dosed, pharmaceutical grade, and physician formulated. Check them out at transcriptions.com. Also brought to you by Health Optimization Medicine and Practice, a nonprofit organization that's training doctors and practitioners on how to optimize health rather than treat disease. Check them out at homehope.org.